Genes do not control biology. Why? Biology still has control even if there are no genes in it. So something else is involved with controlling biology. So I'm back there culturing these stem cells, and most of you have heard of stem cells like this hot topic. I was culturing stem cells back in, my first cultures of stem cells were 1967. So that was, a long time ago kind of stuff, right? And they taught me so much information that I actually had to resign my position because I realized what the cells were teaching me in the cultures were not what I was teaching medical students. And the discrepancy, I just couldn't live. It's like I can't go in there and tell lies that genes control life when I know they don't. What was I working on? These special stem cells in your body. What are stem cells? Stem cells are the equivalent of embryonic pluripotent cells, meaning this. In your body right now, there are cells scattered throughout your body that are embryonic cells. And what's their function? Well, if you damage something or you need to build something back up, these cells then can contribute to the growth of whatever you're building or replacing in your body. All of us have stem cells. And what was interesting to me, and I just love it because it's a joke, that we are now pushing for stem cell research. Why? Well, apparently we have stem cells, but they don't work, and so we have to give them to the pharmaceutical company, and they will figure out how they make them work so that we can take drugs to make them work. And the point is, do you think God gave you stem cells to wait for the pharmaceutical industry to figure out how to make them work? The issue is you have them. The question is, why aren't they working? Well, part of it started to come through in my understanding about the cultures. I cultured these very identifiable, very specific stem cells called myoblasts. They're muscle precursors. And what I would do is isolate these single cells from muscle. And then I plate them in a culture. And you see these little dots behind these strappy looking things? See the little dots all around here? Those dots are these cells. When these cells start to grow into a colony, they ultimately at some point differentiate and form these very large muscle cells that actually contract in the petri dish. And they are derived from these single cells. And these are committed cells committed to form muscle. At least that's what I was working on. And at one point I started to change the culture environment a little bit. And as I changed the environment, these myoblasts, which gave rise to these cells, changed and became these kind of fibroblasts and gave rise to these cells. And they can form something completely different. They form bone matrix. And I said, wait, if I take the same cells and change the environment, they form bone. And then I found out if I change the environment a little bit more, the same cells would form fat cells. And then all of a sudden I started to realize, what's controlling the cells? They all have the same genes, but in this environment they form muscle, in this environment they form bone, in this environment they form fat. What controlled the cells? It wasn't inside the cell, it was the environment that activated the gene expression. Why is this relevant? Because if I took my petri dish and put it in an optimum growth environment, the cells grow and they flourish and divide and become healthy and do everything. If I take the same dish and move it to a less than optimum culture environment, they start to get sick. The dividing starts to go down. They don't express their fate anymore. They begin to die. And if I take them from that bad environment as they're beginning to die and put them back into the good environment, they recovered their life again. What's the relevance about this? The simplest conclusion is this. You are a petri plate with 50 trillion cells in it. Put it in a good environment and it will grow. Put it in a bad environment and it will stop growing. All of a sudden, it wasn't your cells that were pre-programmed to be sick or not. It was the way you responded to the environment that controlled the expression of the cells. So where we were giving credit to the genes, we now have to look and say, wait, the genes are important, but the control comes from the environment. So I write this up in a paper back in 1977, and I write a fine structural analysis of normal. I didn't have another word at the time, modulated. They changed their behavior. I couldn't figure out what it was. Tried to talk to my friends. They were all into genes. They look at me and go, we don't have time for that. And after a while, I realized it was such a deaf ear that I walked out of the university because I thought, if you can't see how important the environment is, then I can't stay here and do this. And at some point, I came back into biology again because Stanford was open enough to consider weirdos like myself. And it was wonderful because then I also had an opportunity to be in amongst the likes of people like Bill Tiller and Carl Pribram because Stanford was a very unique environment to offer this. And so I do this work at Stanford, working with human blood vessel cells, same thing. And I get this, I get the cover of Differentiation. And I really wanted the cover of Rolling Stone, but And what do I get? A microvessel, that means blood vessel, capillary endothelial cells, transdifferentiation. New word. Because I figured, hey, Stanford, you can't use a cheap word like modulated from, Wisconsin. So I used the big $10 word, transdifferentiation, meaning that if I change the environment, the cells change their fate. So I do this for a while. In about 1992, after I repeated the experiments and published them, and I realized at some point they're still interested in genes and they don't want to talk about this, I leave and decide I might as well just talk to the people directly. So I end up writing this book, which took a number of years to get the science so people could read it, because the first time I wrote a manuscript, it was so scientific. They were so embarrassed to tell me they couldn't understand a damn word that I wrote. They just avoided me like the plague. So I finally got it down into human words here. And what do I write? Now, here's what I write. I want you to note, in chapter 2, I lay out the scientific evidence to show you genes do not control biology. I also introduce you to the exciting discovery of epigenetics. A new field of biology is unraveling the mysteries of how the environment influences the behavior of cells without changing the genetic code. And so what do I call chapter 2? Well, I call it, It's the Environment, Stupid. And basically that's a takeoff from the Democrats who were saying it was the economy, stupid. And I thought this was a funny title. And yet when I wrote this book, and most people, the conventional people, looked at me and said, Bruce, what the bleep do you know? And I had to stand there and say, yeah, but all this is still, I support this, the experiments support this, and all this was really great. And then, guess what happened just 2 months ago, or 3 months ago now? It was actually May, so it's even maybe a few more months ago. Issue of Nature, one of the most prestigious journals in the world, Nature. There's an article. What's it about? Stem cells are engaged in a constant crosstalk with their environment. Biologists are fast realizing. What do you think the article is called? I love this. It's the ecology, stupid. I rest my case, you see. It took them some years, but they're getting there. So the question is now, what do we learn from all this? Well, what we're going to learn is how human cells work. How do they work? Well, let me explain this, and this is where I have to give you the scientific facts enough so that the conclusions make sense, that I'm going to give you in a few minutes. How do cells work? Well, the easiest way to show you how a cell will work is use the transmission from my car. And the reason, because a cell is made out of parts, like a transmission. But this is an important diagram because it's called an exploded diagram, and it shows two different things. A, it shows all the individual parts that go into making a transmission. All the individual gears, nuts, bolts, washers, bearings, cotter pins, all the different parts. But it also shows this. The parts aren't just thrown into the transmission. The parts are grouped into assemblies, and the assemblies interact with assemblies. So basically it says there's an order going on inside here. Well, the truth is this. The cell is like the transmission. The parts are made out of proteins. So when I look at a cell and I open it up, I got all these parts, all different shapes, over 100,000 different kinds of proteins. They don't look like man-made parts because they're these organic parts. They're nature. And they don't look like man-made parts, but they're the equivalent of steel because they're the same parts that are essentially in each one of you. That's why I can exchange parts with each other. And that's why people can share organs and things like that. They're almost the same. And so that the cell is like a transmission, and the gears are made out of the proteins. Well, what is responsible for giving each gear its so unique shape? And there are over 100,000 of them, as I said. And the answer to that, let me use a protein that is in every one of you right now. If it isn't, you're not here. This protein is called calmodulin. You need it for your life. This is one of over 100,000 different shaped proteins. Well, it looks like a whole bunch of foamy soap bubbles the way the biochemists illustrated right here. Actually, each of these little bubbles represents an atom. And so this is the complex molecule. Now, what's interesting about this is calmodulin has a unique shape, makes it different than all the other proteins. But while it looks very soap bubble-like, it's the equivalent of steel for this reason. Take calmodulin out of the person sitting next to you, put it in your body, it will work in your body the same as yours because they're almost all exactly the same. The question is, where does the shape of a protein come from? And so to show you that, really, we're going to talk about what is called the backbone. That there's a backbone that gives each protein its unique shape. And what I'm going to talk about right now is interesting is this. This is a very rare opportunity where you can say that all proteins are built on the same pattern. So now when I say this in a couple of minutes, I can describe the entire field of 100,000 proteins. It goes like this. All proteins are linear strings of beads. The beads are called amino acids. There are about 20 different kinds of amino acids. You can go to the health food store. When you're buying amino acids, you're buying the building blocks that make up the beads. Well, the important part about this is what makes each protein unique. And the answer is this. Each one has a very specific and unique characteristic alignment of amino acids of the 20 different ones making them up. If I did this right, I would have 20 different colored beads, but I don't have that many different ones. But the point is this. The sequence of the amino acids is unique for each different protein. But then if you look at it, there's a little misleading thing about this model because it suggests that each protein looks like a strand of spaghetti. That's incorrect because each amino acid is not the same shape. It doesn't, this is the failure of this model, so I go to this model. And I say, let's say there are 20 different amino acids, which there are, and each amino acid has a unique shape that says there are 20 different shapes. It's like a giant Tinker Toy set, okay? I'm going to show you 3 different pipe fittings. Yeah, I'm standing in the light. Okay, 3 different pipe fittings, each with a different shape. First pipe fitting, the yellow one, is straight. The second pipe fitting is red, and it's got a 45... Can you see that? I'm looking direct in the light. Can you see it? Okay, this is a 45-degree bend on this one. And the third amino acid that I'm going to use has a 90-degree bend. So this is 3 out of 20 shapes. That's all I need you to know. There's a lot more shapes. But they are like poppet beads because they plug into each other. This is called a peptide bond. So I bind it together. And guess what? Now I'm creating a chain. But no longer is that chain that flexible, like poppet bead chain. Now there's a structure like a hard, rigid backbone that gives the shape to this particular assembly of proteins. So as I'm assembling my amino acids into a protein backbone, this is the shape. And all of a sudden you say, it's like the poppet beads because, look, one color followed another color, another color, another color. It's a chain. But because they're unique shapes, it's not a flexible chain like this, but it has a unique characteristic shape. Is that evident to you, I hope? Here's the next point I have to ask you, though, very clearly. Do you recognize this point? This is the real critical one. If I disassemble this and reassemble it in a different sequence, using a different sequence, will I get the same shape, backbone, yes or no? Okay, well then you've got the fundamental understanding of why each of the proteins is unique and different. Each one has a different assembly of these subunits called amino acids. So basically, the amino acids join up in a backbone, make the proteins. The proteins become like gears in that transmission. Well, let me give you this picture of a watch. Can you imagine in your head, when gear A turns, it turns gear B, and when gear B turns, it turns gear C? Can you imagine that? Now I say, well, wait. How about if I blew proteins up big enough to be the size of gears? So I lay on top of this, instead of metal gears, I put protein gears. Okay? Now, question though, in your head, can you imagine when protein A turns, it turns protein B, and when protein B turns, it turns protein C? Can you imagine that? Well, then I say, okay, get rid of all the man-made parts. Boom. What do we have left? A machine. It's made out of proteins. In your head, can you imagine when protein A turns, it turns protein B, and then protein C is turned? Can you see that? Well, that's like, remember in the transmission I said there are assemblies? Assemblies of proteins that interact to carry out a function are called pathways. Now, I'll throw a word out, see if you can figure this out. What is a digestive pathway? It's a process of digestion carried out by what? A series of proteins acting as gears, one turning the other, with the result digestion, right? Okay, you got that one. Here's a better one. Respiration pathway. What is that? A group of proteins assembled together when they interact, do respiration. Okay, and the last group of people who have gotten that yet, here's the last one. Muscle contraction pathway. A group of proteins that assemble and cause okay, what's the nature of the cell? It's got all these assemblies of proteins carrying on the function, but here's the problem. In order to have function, it's got to move. And movement is where life comes from. Life is movement. It's characterized by movement. Here's the point. If the proteins don't move, you have a cadaver. That's what a dead person is. A person who's just all proteins and no movement.
If you like Succeed with Knowledge, subscribe and give us a thumbs up. And thanks a million. Cheers.